Laboratory-based biomarkers to improve causality assessment in drug-induced liver injury. By Daniel Antoine. Drug-induced liver injury causality assessment and the need for new biomarkers. Drug-induced liver injury, or DILI, represents a major medical concern associated with significant patient morbidity and mortality. Within drug development, attrition caused by DILI occurs in all phases of the pipeline, from preclinical testing to clinical trials to the marketplace. DILI is largely categorized into two major forms, intrinsic and idiosyncratic. In cases where the frequency is high in either animal species or in humans, DILI is considered intrinsic in that it is assumed to result from direct hepatocellular damage. Idiosyncratic DILI occurs rarely in susceptible individuals exposed to therapeutic doses, but can represent a serious adverse drug reaction. It should be considered in patients who have deranged liver function tests secondary to the administration of a potentially hepatotoxic agent. Idiosyncratic DILI is unpredictable because of the complex nature of the mechanisms of the disease. Currently, the diagnosis of DILI is based on exclusion. The exclusion of other differential causes and an understanding of the temporal relationship to the administration of a potential hepatotoxin represent the cornerstone for establishing a diagnosis of DILI. For causality assessment, different methods, such as the Roussel-Uclaf Causality Assessment Method, or RUCAM, or a consensus-based expert opinion approach, are two main mechanisms used. Despite relative strength, each mechanism is also associated with a number of limitations. A major component of these causality assessment methods relies on the use of a small, but routinely measured, panel of markers, such as aminotransaminases, that is, alanine aminotransferase, or ALT, and aspartate aminotransferase, alkaline phosphatase, and total bilirubin. Challenges associated with these current biomarkers are that their elevations are not limited to human DILI and can occur in other forms of liver disease and also in cases of cardiac or skeletal muscle damage. The elevation of these markers is often delayed in nature, and there is a limited understanding of their kinetics in the natural course of this disease. Significantly, these currently used biomarkers lack a fundamental mechanistic basis. Moreover, they do not allow prognostic information because circulating levels do not reflect the actual extent of hepatic damage and can occur frequently in clinical trials without degenerating into serious hepatotoxicity. Therefore, new laboratory-based biomarkers or tests that can be applied to diagnostic or prognostic algorithms that predict the clinical course of DILI would greatly facilitate the management of acute DILI and lend support to already used causality assessment methods. The specific diagnosis of DILI is hampered because there is currently no specific or objective test. With the exception of acetaminophen, or APAP, protein adducts, there is no specific non-invasive diagnosis of DILI. In addition to this, a variety of in silico-based methods have been developed to understand susceptibility to DILI at the level of the drug by correlating physiochemical properties to the severity of injury induced. Therefore, there has been considerable effort to identify and develop new biomarkers that can inform the mechanistic basis of DILI and provide potential measures for patient management. See Figures 1 and 2. Because of the multifactorial nature of DILI, it is clear that no one-size-fits-all biomarker will suffice. Recent evidence from clinical and preclinical studies have described the development of such a panel-based approach that includes both circulating biomarkers and cell-based screening methods to rule in or out liver injury, provide early detection of liver injury, and enhance the mechanistic understanding, prognostic information, and definition of the causative agent. Candidate biomarkers and in vitro screens. Idiosyncratic DILI is a rare event. Therefore, the initial question posed by the investigator is, where do we start and with what drug? The majority of published data in the literature reflects clinical and preclinical studies of APAP-induced hepatotoxicity. APAP hepatotoxicity is a major medical problem, and 50% of cases of acute liver failure are attributed to this drug. Therefore, this patient group represents an ideal place to start to test the performance of novel biomarker test candidates. See Table 1. Recent attention in the literature has focused on the development of microRNA biomarkers of liver injury. Promising candidates have yielded microRNA 122 or miR-122. miR-122 expression is exclusive to the hepatocyte, and it represents 75% of the total hepatic miRNA content. Clinical, preclinical, and in vitro studies have demonstrated that miR-122 elevations can be detected earlier than changes in traditional markers of hepatotoxicity or cell death. The outcomes of these studies are twofold. 1. miR-122 over ALT elevations can assist with the diagnosis of liver injury, and 2. The increased sensitivity of this biomarker in a temporal sense compared with currently used indicators can be used to identify liver injury earlier and aid treatment and risk stratification. It is thought that the mechanistic basis of DILI can greatly inform potential therapeutic strategies and aid prognostic determination. The quantification of circulating levels of the intermediate filament keratin 18, or the caspase-cleaved keratin 18 form, has been demonstrated in liver disease, and the relative abundance of these markers, reflecting total cell death and apoptosis, has been informative for monitoring cell death mode dynamics during APAP hepatotoxicity and aiding in prognosis. Given the importance of the mitochondria in regulating cell death mode, recent studies have demonstrated that monitoring circulating biomarkers associated with this can inform further on the mechanism of hepatotoxicity in human and mouse and also provide enhanced prognostic information. Activation of inflammatory pathways is a hallmark of many acute and chronic liver diseases. These represent important mechanistic steps in the development of injury or repair that regulate patient responses and outcomes. The identification of key mediators of these events has also yielded potential candidate biomarkers and targets for therapeutic intervention for DILI that are inherently linked to the mechanism of the disease and can provide enhanced prognostic information. High Mobility Group Box 1, or HMGB1, is an endogenous danger signal that sits at the intersection between sterile and infectious immunity and is vital for linking cell death to inflammation. The conditional knockout of HMGB1 and its targeting with monoclonal antibodies protects mice from drug-induced acute liver injury. Specific HMGB1 isoforms, such as its acetyl form, have been demonstrated to inform on patients spontaneously surviving or requiring liver transplantation secondary to APAP overdose. Inflammatory responses serve to restore normal hepatic architecture and promote regeneration. One key mediator, colony-stimulating factor 1, or CSF1, regulates these events by promoting enhanced macrophage functionality and phagocytosis of cell debris. Its circulating levels have been demonstrated to follow an opposite trend to acetyl HMGB1, which is elevated in patients surviving APAP overdose and lower in those dying or requiring a liver transplant. Therefore, its measurement in parallel with acetyl HMGB1 represents a potentially powerful combination of tests. A significant challenge in determining causality assessment in patients with liver injury is associated with polypharmacy, especially in cases in which several drugs are known hepatotoxins. Recently, a new in vitro test system, MetaHeps, has been demonstrated to provide added value for the determination of the causative agent in cases of polypharmacy. In this system, patient-specific monocyte-derived hepatocyte-like cells with inducible cytochrome P450 and active urea production are more susceptible to cytotoxicity induced by the suspected drug compared with cells derived from healthy donors. Moreover, these cells have been demonstrated to outperform RuCam scores. Concluding remarks. The development and maturity of national and international prospective DILI registries over the past 20 years has now made it possible to determine the utility of these promising candidate biomarkers in cases of idiosyncratic DILI. Recent collaborative efforts and results between consortia, such as the Safer and Faster Evidence-Based Translation, or SAFE-T Consortium, the Predictive Safety Testing Consortium, or PSTC, the Drug-Induced Liver Injury Network, or DILIN, and the Spanish DILI Registry, combined with preclinical and clinical studies of APAP overdose, has recently led to regulatory support from both the European Medicines Agency and U.S. Food and Drug Administration for their more systematic use and further qualification. The further development of these markers in well-controlled Phase 1 studies and trials designed to test the ability of the biomarker to guide patient care should remain the focus of new efforts. An exciting proposition and direction for future studies to aid causality assessment in human DILI would be a combination approach of predictive in vitro cell systems with the aforementioned novel mechanistic and hepatic selective prognostic biomarkers to be used in parallel with current tests. Advances in software, databases, and data visualization approaches such as DILISIM, LiverTox, available at www.livertox.nih.gov, and eDish, which is Evaluation of Drug-Induced Serious Hepatotoxicity, may further realize the promise of these novel approaches. For more information on clinical liver disease, please visit cldlearning.com. Thank you.